Hey guys, welcome back. Now, last lesson we were talking about transcription and translation, and just a general overview of protein synthesis. But this whole lesson is about gene expression. So to conclude this chapter, let's talk about how that protein synthesis is regulated in a general lesson called gene regulation. This begins with the first part, which is pre-transcription, or before we start creating your mRNA. Now, you may have seen this diagram before. It's DNA with their histone molecules, or wrapped around their histone molecules. But before we talk about those histone molecules, let's talk about transcription factors that regulate how RNA polymerase has access to the DNA. Now, we have a lot of words here, but just to simplify it, let's start with the activator and enhancer. So our enhancer is just this sequence of DNA. And our activator is able to bind to this enhancer part of the DNA. And it activates and enables RNA polymerase to access that area. Now, you also have an area called an insulator. And that is this area of DNA that I've marked in light blue. And the insulator stops it. So it doesn't allow the RNA polymerase to access the promoter, which would allow the RNA polymerase to start producing mRNA. So your insulator is binded by a CTCF protein. So both the activator and the CTCF protein are both proteins. And now you may have seen that I wrote CCTC binding factor. And that's because in all insulators, you have a CCTC sequence that is found in the DNA. And this kind of marks that insulator, and that's where the CTCF protein binds to. And so when it binds to this insulator, it stops the RNA polymerase from accessing the promoter. But we have these methyl groups that can be found or attached to the insulator so that it blocks the CTCF from being able to access that DNA and allowing the RNA polymerase to access the protein or access the DNA. So just a summary. You have your activator and enhancer. One is DNA, one is the protein, and it enhances or activates the ability of the RNA polymerase to reach the promoter. Then you have an insulator and the CTCF protein, and these do the opposite, so they stop the access of RNA polymerase to the DNA. And then you have methyl groups, which regulate the insulator and allow the RNA polymerase to access the promoter, and the promoter will be found later down in that strand. So we've talked about pre-transcription as far as transcription factors, but there's also a factor called epigenetics. And this is something we actually talked about before. So you have these histone molecules, which I've drawn in yellow, and these histone molecules have DNA wrapped around it because they are charged, and DNA is charged. So DNA is obviously attracted to these histone molecules. And when the DNA is tightly wrapped around the histone molecules, it's actually harder for RNA polymerase to access those sequences. But if the DNA is more loosely wrapped, then the RNA polymerase has easier access and it's easier to create mRNA. And so this is just pre-transcriptional regulation through epigenetics. And it's a new field that a lot of research is being conducted in. Now that we've talked about pre-transcription, let's move down our DNA to operons. And operons now are common in prokaryotes. So the example that I was talking about before with transcription factors, those are found in eukaryotes. But operons are generally found a lot in prokaryotes. Now, our operon has three parts. You have a promoter, an operator, and a structural gene. It also has a suppressor, but we'll talk about that in just a second. So you have your promoter here, you have your operator here, and your structural gene here. So what the structural gene is it creates the actual, let's say, enzyme that you're trying to create. Your promoter is where the RNA polymerase binds to, or so that it can produce the mRNA that's coded for in your promoter. But in between these two steps, you have your operator, and your operator has a area called, or has a protein called the suppressor. And what the suppressor does is it's attached to the operator and blocks the RNA polymerase from being able to access the DNA until you have some type of protein or some type of trigger that removes the suppressor from the operator. And in this case, let's say it's a substrate. Let's say it's sugar that an enzyme is coded for in your operator. And so because the sugar is present, your suppressor leaves and the mRNA is produced. And this is actually kind of helpful for the bacteria, because this way it only produces that mRNA when it's necessary, when that substrate is present. Because if it's not, and it's just unnecessarily creating those mRNA sequences, it's wasting energy. So this is kind of good for this type of regulation is good for bacteria, and in general, just regulation helps preserve the resources of the cell. We've talked a lot about pre-transcription and during transcription, but we'll conclude this lesson with post-transcriptional factors and post-translational factors. And so specifically for post-transcription, you have something called RNAi, and it's another RNA. And specifically what it is it interferes, or it binds with a mRNA that, let's say, the cell does not want it to be produced. So for regulation, the RNAi binds with your single-stranded, so let's say this is your RNAi, it binds with your single-stranded RNA, creating double-stranded RNA. Now, double-stranded RNA, the cell will react to this because usually there is no double-stranded RNA. And so it'll cause the original mRNA to be destroyed, so you won't have it anymore. And that's how RNAi kind of functions in a more general explanation. This is a kind of complicated process in some cases, so we'll be learning more about this later on. Now, post-translational regulation is with the protein, because you've already created the polypeptides after translation. And so what happens is the protein can be affected by the pH, environment, it could be phosphorylated, or it could bind to another protein that would suppress it. And so this can also factor into regulation. And so you have all of these processes that factor into gene regulation. And this kind of concludes our entire chapter on gene expression. Hopefully you've learned a significant amount about this. And if you have any questions, feel free to ask in the comments below. Thank you.